serial killers of the immune system, also known as CTLs or cytotoxic T lymphocytes.
Their full history dates as far back as the 1960s, when researchers first started to get a hint that there might be a cell that was killing other cells. In the 70s, researchers found one cell type that did this, and they gave them the name cytotoxic T lymphocytes. Lymphocytes meaning cells in the lymph, T stands for thymus, from where they developed, cyto meaning cells in general, and toxic, well, you understand. The 70s was also when CTLs got their first break on the big screen with Microcinematography. Yes, this was their first photo shoot in black and white. And then in the 2000s, almost 30 years later, they got their first movies in fluorescent colors. Here is one CTL in red, delivering the kiss of death to a cancer cell in green. See how the red CTL is pumping its toxic payload into the cancer cell, and the cancer cell membrane ruffles and the green fluorescent protein leaks out of it, leaving a clear, translucent cell corpse behind.
Results like these were really exciting at the time,
and led to mathematical estimations that, at their best, CTLs might be able to kill up to 5 cells per minute or even hundreds of cells per day.
And so, let's give them CARs to use them in cancer immunotherapy. Yeah, I said CARs, but not these CARs. CAR stands for chimeric antigen receptor, and this is how it works.
If you have a type of cancer cells with a protein on their surface that could be used to flag them as different from normal cells, a chimeric antigen receptor, or CAR, could be designed to target that flag. CTLs engineered to have this CAR, which is now called a CAR T cell, will be able to recognize the cancer cell flag and pump toxic proteins into that target cell to kill it. CAR T cells designed for B-cell cancers, which has the classic B-cell marker CD19 on their surface, has been tremendously successful, with a consistent 70 to 80% response rate over the course of various clinical trials since the early to mid-2010s. And one second-generation CAR T cell was estimated to be responsible for the death of more than 1,000 chronic lymphoblastic leukemia cells.
So all this is looking really good for blood cell cancers or hematological cancers. And so scientists are starting to try to make designer CAR T cells for solid tumors, but the results there are not looking as promising.
Part of the reason is that a lot of these cells seem to get stuck in the borders of the tumor, or their killing capacity is actively being held back by surrounding immune cells in the local tumor environment. So there's a huge amount of effort going into tackling both of these problems.
Was that all entirely unexpected? Well, yes and no. If you look back at papers published in 2008 and 2009, you get some idea that these cells may not be as good at killing cancer cells that form solid tumors as they would be blood cancers. 2008, isn't that just 2 years after that really promising study? Exactly. And even more intriguing is that the same cells that were used as the blood cancer model in 2006, in 2008, when these cancers were put into a solid tumor models, they became a lot more resistant to CTL killing.
Compared to when they were free-floating, where each CTL could kill a cancer cell in a few minutes, when they were in a solid tumor, it took up to 6 hours for one CTL to kill one tumor cell, and frequently a few CTLs needed to work together at this. And then look at this 2009 study. Researchers show that human CTLs are also abysmally bad at killing melanoma cells, which are the prototypical solid tumor models. Again, where lymphoma cells are killed within minutes at 1 versus 1, killing melanoma cells required hours and several CTLs working together.
So by this estimation, a kill count of hundreds of cancer cells per day just doesn't sound very realistic. But fear not. CTLs are not the only serial killers of the immune system.
You see, CTLs are also called CD8 T cells because they have the signature CD8 protein on their surface. This distinguishes them from their sisters, the CD4 T cells, which don't have any killing capacity of their own. But if we move up a bit in the immune cell family tree, we come to an aunt, the natural killer cells, or NK cells. Now, these gals are some professional hit-and-run assassins. whereas CTLs could actually be caught in the act because each CTL will be filling up their target for 30 minutes to several hours, sometimes ganging up on their targets and sometimes coming back to the same target over and over again. NK cells, on the other hand, are much more efficient. It often takes them less than 5 to 10 minutes to perform a kill, but this also makes their assassination act much less easy to catch on camera.
Of course, NK cells have their own drawbacks as well.
One study that came out in 2022 found that NK cells were very efficient in the first 24 hours, but over time that efficiency seems to vanish.
Nevertheless, the fact that they are so efficient at their prime is still good enough for them to start getting their own CARs. For the future of cancer immunotherapy, we might be looking at CAR T cells or CAR NKs or a combination of both, depending on the properties of the individual cancers. And for now, whatever we learn from one killer cell type could certainly be used to improve upon the other.